Hello, Bio4. Let's start part 3. And we start part 3 talking about the glial cells. And these are the support cells, these are the babysitting cells, remember, that are going to take care of those neurons. So the ratio of how many neurons we have and how many glial cells we have is alarming. There is about 50 glial cells to each neuron. So these are a lot of babysitters for these cells that we want to stay alive. I'm calling it no neuron left behind because we really do have to take care of them. So it's a really nice support system. And if you look at this picture here, it's way too crowded for you to understand, but there's lots of cells here that matter and that we're going to look at. There's these yellow ones here that are lining, ependymal cells. There are these star-like green ones. That's an astrocyte. We're going to look at these microglial cells, and we're going to take one at a time. What you need to notice here in this diagram is that the cells that are getting taken care of are these, right? So here is a neuron all the way down here, and here's another one, right? And all the other ones are these babysitting cells that are going to do all sorts of things to make sure that their environment is just perfect so that they survive. Okay. Let's take one at a time. And the first one is an astrocyte. So astrocyte looking like a star, astro, and it definitely looks star-shaped here, okay? These are really awesome cells. They're amazing. So look what it does. This looks like a cell body like any other one, but then it's extensions here. Look, it's dendrites. What do the dendrites do? They come around and they wrap, right? They're going to wrap around here the axon or the dendrites of this cell, this neuron that they're protecting. And not only do they do that, but then they're going to reach out on the other side here and they're going to wrap around, what's this red one? This is a vessel. This is a little capillary. So at one end, it's wrapping around the neuron. At the other end, it's wrapping around a capillary. What in the world is it doing? Why is it doing that? Well, my description for this is that they're the king's tasters. You don't want to kill this cell off by something toxic that has come into the body. So what do? You filter everything. You know the story of the king's tasters? Everybody wanted to kill the king. Not everybody, but some. So they would try and poison the king. So kings, this is years and years ago, would have a taster. And before he sat down to his meal, there would be a taster who would first eat the meal. And if you dropped dead, well, then the king wouldn't eat it. And what a job, huh? Great job. So here, the astrocytes are kind of the king's tasters. So they will absorb nutrients from this vessel here, take it into the cell, take it into the body, and then filter it and pass it over now to the neuron. So if something were to damage the cell, that passage is not going to happen on to the next one. So truly the king's tasters. If this cell were to die, at least I've saved the neuron and not the glial cell. So these marvelous glial cells are astrocytes. Let's see, description, they're star-shaped, that's right, hence the name. They're able to brace a neuron. Yeah, they're bracing a neuron. They form the blood-brain barrier between capillaries and neurons. So that's what the blood-brain barrier is. I don't know if you've heard of that. Sometimes we have pharmaceutical drugs that can't reach parts of the brain because of the blood-brain barrier. So you give this medication to an individual and it's in here, right? It's in this vessel and this astrocyte takes it in and says, I'm not passing this on. This is not a good thing. And I'm going to keep this medication here and not pass it on. It's a filter. And so some of the, like dopamine, for example, is one of those. When we give artificial dopamine to someone with Parkinson's disease, it has to be a slightly different form of it. It's called L-DOPA. And that different form is not recognized by the astrocyte, and the astrocyte passes it on. But the regular form of dopamine, it recognizes and it says, not passing that on. So that's the blood from here, brain barrier. And the astrocytes are the ones that are responsible. They completely control the chemical environment of the brain. They also store sugar. I think I've written that. Yeah. So they also supply glucose to neurons. They store sugar. So your brain is never going to run out of sugar. I remember being told that when you had an exam or you had an SAT or an important t-test or something, that you really had to have breakfast because you had to feed those brain cells, right, for this test. That's not true. These astrocytes are full of glucose, and if the level of glucose in the brain were to dip a tiny bit, these astrocytes are releasing that sugar to the brain. So if you skipped breakfast before your test, it's okay. You're going to be fine. They also secrete nerve growth factors, and we know that because in an individual with a stroke, for example, where there's been damage to some axons or dendrites, we see lots of these astrocytes in the vicinity trying to remedy this problem. So these growth factors are going to promote that neuron growth post-stroke. They're also a cleanup crew. They clean up neurotransmitters and any potassium ions. So potassium ions can accumulate in the tissue fluid, and astrocytes will sort of mop that up and remove it so that that tissue fluid is not ridden with too many of these potassium ions. Where are they? They cover the entire brain and the gray matter. They're everywhere. They're really fabulous cells. And if when we keep studying them, we see that we learn more and more is discovered every year about them. Wonderful astrocytes. Okay, what else? That's cell number 1 is a glial cell, an astrocyte. Let's get number 2. Number 2, oligodendrocytes. Oligodendrocytes. So what they do is they produce the myelin sheath. What's a myelin sheath? The myelin sheath is what wraps around an axon of a neuron to protect it, to make it not lose ions. It's kind of like the plastic rubbery coating over a cable of yours or something, a cable or plug-in from a lamp. Okay, what you're doing is you're trying to insulate that so you don't lose some of those electrons. So that's what the myelin sheath does. It's a sheath that covers the nerve fibers and doesn't allow the loss of those ions. So look at this cell here. It's another miraculous one. Here's a cell, and then this is still part of the cell that's wrapping like a jelly roll around the nerve fibers in the vicinity. Amazing. Insulator, oligodendrocyte. One more picture of the oligodendrocytes. This shows the cell body and then how it has an extension to jelly roll wrap around this one. Another extension here that comes to jelly roll wrap around another nerve fiber. So they can send out these arms and form these wrappers. Now those oligodendrocytes are very, very similar to Schwann cells. They do exactly the same thing. They jelly roll and wrap around an axon. But the only difference is that these are in the peripheral nervous system. Oligodendrocyte within the central nervous system, Schwann peripheral. Okay. And there's the jelly roll fashion. The nodes of Ranvier are the gaps. So it'll wrap around here, gap. And then we have another wrap around, and then gap. And then another wrap around. Oh my gosh, it's so hard to draw with this. And then, so anytime you have a gap like this that is unmyelinated, that is a node of Ranvier. Okay, we've gone through 3 of those cells. Schwann cells recently, there's this new discovery that they've been they've seen some of these on the border of the epidermis and the dermis. So what they're doing there is they're sensing and conveying any thermal and mechanical sensitivity. So anything that's too hot, too cold, or too abrasive is going to receive that signal through these Schwann cells. I see a spelling error here, and then communicate with the neurons. Okay? All right. These are, this is a picture of that. So you'd have this Schwann cell here coming up to the surface, and these then are They're responding to a mechanical signal. I'm going to keep going so that I can finish the glial cells. The microglia are the cleanup crew. So they too look a bit like a star. They've got these long extensions, microglial cells. They look like little spiders, and they are the janitors. They will dispose of debris. They're concentrated in areas of damage. Again, if you see a stroke, you're going to see lots of these, trying to clean up any kind of blood that's bled, any kind of damaged cell that's sitting there. They're going to be able to come and eat that up and be able to remove it from that site. Microglial. Ependymal cells. Ependymal cells are liners of cavities in the brain. So you didn't even know you had cavities in the brain. Did you know that you had holes in your brain? Well, you do. You have spaces in there that are full of cerebrospinal fluid. And those spaces need to get that cerebrospinal fluid because we produce about 2 cups of it a day. And you need some specialized cells that are going to do that. They're going to produce that cerebrospinal fluid and dump it into these cavities and then make it go down the spine, the spinal cord. Those are ependymal cells. This cerebrospinal fluid, let me show you, bathes your brain. So you would see it here with these arrows all the way around the brain, around all aspects of the brain here, the cerebellum, and then all the way down the spine. 2 cups of it, you guys. You produce a lot of this, bathing your brain. 2 cups a day. The last one is the satellite cells. Satellite cells surround cell bodies. We don't know a whole lot about it, but it's these little hats here that we can see on the actual cell body. We think they might regulate chemical environment. We're not sure. Maybe they provide some sort of electrical insulation. We're still uncertain exactly what these satellite cells do. So we're back to that slide that has all of them in one place. But I'm going to stop this now. We'll start the next one with this slide and review each one of those on their own. Thanks for listening. Bye.